So next disease we'll talk about is tularemia, and the causative agent of the disease is Francisella tularensis. So bacteria belongs to separate species, Francisella, and actually nowadays we have only one species, Francisella tularensis, which is divided into 3 subspecies. Please take a look on that microscope view. You can see that it is gram-negative bacteria, but it's not really obvious is it a rod-shaped bacteria or coccus. So bacteria is really very small, and it should be rod-shaped bacteria, but because of unique complex growth requirements, bacteria very poorly grow in couch medium, and actually in slide it's looking like a coccus, although in clinical samples it's more looking like a rod-shaped bacteria. Bacteria is capsulated, non-motile, non-sporin, and belong to obligate aerobic respiration type. Bacteria is known as facultative intracellular parasite, means that it can grow in couch media without any cell. But in clinical samples, more often it is located inside host cell and acting as an intracellular parasite. Bacteria is one of the most contagious. About 50 bacterial cells is enough to cause a disease when inhaled, or even can penetrate through the skin. Bacteria as well belong to the group of potential biological warfare agents, and in routine laboratory practice it's classified as a biosafety 3 level. As we said, we have 3 subspecies of bacteria, but as well those subspecies are known as biogroups or types. So Francisella tularensis subspecies Tularensis is known as type A or biogroup A, and it is the most dangerous subspecies of that microbe, because it causes a very high mortality rate in animals, and in human diseases acuity is more severe with poor outcomes. Glad for us, bacteria is very rarely isolated in our continent, and that type is prevalent in mainly USA and Canada. Second subspecies is Holarctica, as well known as biogroup B. That strain is less virulent for human and for animals as well, and that type is circulating in our regions, in European countries, in Asia, and in our continent in general. Third subspecies is very rare subspecies. It is Media asiatica. It is isolated only in some countries, in some regions of Central Asia, and very low incidence of disease in human, and usually they are very mild, and bacteria is rarely registered and reported. One more species of Francisella is Francisella novicida, and some scientists say that actually that microbe should be classified as a 4th subspecies of Francisella tularensis. Till the moment it is a still separate subspecies, but for human pathogenic properties is very low, and few accidental cases were reported in people with severe immunological disorders, and very low data is available to report and to discuss the species. So as we said, microbe is fastidious, means it needs unique nutrient requirements, unique culture medium. It requires several amino acids and several other compounds for growth. It's better to enrich air with CO2, and anyway you will obtain some visible growth only in a few days after inoculation, and you can take a look on that picture with culture medium. You see that anyway colonies are very small, they are buttery and usually merging one with each other, looking like some cream on the petri dish, and growing slowly. To cultivate Francisella tularensis, it's better to use chocolate agar or chab agar. It is type of blood agar, but used mainly for Francisella tularensis. If you will talk about differential properties of Francisella, it's difficult to study, because bacteria again is very fastidious in the growth requirements, and it's difficult to use some routinely used culture medium, biochemical or indicator culture medium, which is usually used because bacteria will not grow on that culture medium. So usually results of biochemical activity again will be obtained only in 3 to 4 days, and it's of course not suitable way to study and to confirm Francisella etiology. So for identification of bacteria, usually we're using some skin tests, allergic tests to detect hypersensitivity in ill or suspected subject. We can use reaction of agglutination or PCR as the modern technique. In some cases, or when other methods are not available, it is possible to use inoculation in susceptible animals such as guinea pig or mice. But biochemical identification and cultural identification is rarely used in medical practice. If we'll talk about virulence factors of bacteria, we mentioned already that it have a capsule, which again helps to resist phagocytosis and activity of complement and other immune factors of immune system of human and animals. And in experiments, removal of that capsule leads to loss of virulence in strains, and that bacteria became not able to cause a disease in human. As gram-negative bacteria, of course, it have endotoxin, lipopolysaccharide. Again, it's not such active as in usual gram-negative bacteria, but still have some activity. It have number of hemolytic enzymes, and as well via antigen is described. So one of the most important properties of Francisella is ability to multiply within phagocytic cells of human. As well, bacteria is able to survive on hepatocytes and can resist activity of lysosomes and enzymes, lytic enzymes of our cells. It can leave that phagolysosome and enter cytoplasm. So it can avoid activity of enzymes by migrating from vacuoles of phagocytic cells. It have number of protective proteins. I will not list them for many of them, but they are difficult for description in current lecture. And main tasks of those genes is first regulation of virulent genes, which cause some factors of pathogenicity. They can inhibit specific receptors and inhibit recognition of bacteria bioimmunity. So Francisella tularensis can hide of our immunity, and our cells will not recognize it as a pathogen. And again, they protect bacteria from oxidative burst of our enzymes, and sometime in phagolysosome bacteria can survive thanks to those proteins. Then bacteria can migrate to cytoplasm and survive even in phagocytic cells. So initially, in very beginning of studies, there were some reports about production of exotoxins, and it was suspected that Francisella can produce some toxic substances. And nowadays, still no exotoxin was identified, and that property is still under the question because maybe it is present, but no genes is found in clinical strains, and in experiments, no other toxins and no secretin factors and culture medium were identified. So if you talk about a disease, tularemia, disease is present again in animals. It is a type of zoonotic infection, and usually rodents, different type of rodents, beavers, squirrels, and hunting rodents acting as the reservoir of the disease. Rarely, disease can be observed in domestic animals and even in water supplies near some farms, but again, nowadays, thanks to good protective measures, animals are rarely ill with Francisella, domestic animals, and usually it is a wild animals, deers and rodents living in regular environment and forests. So, of course, main risk factor is hunting, and hunters usually suffer from that disease. In rural areas, children can be affected because they like to play with animals. They cannot understand is it ill animal or not, and they can touch it accidentally, or they can find dead animal, touch it and take a look to trying to play. And of course, laboratory personnel who working with that bacteria laboratories is affected by disease. Again, microbe is very dangerous, and of course specific safety precautions should be followed, but it is a risk. In mild cases, when disease is not really severe, usually convalescence is spontaneous. People will have few days of some symptoms, non-specific one, and disease will be gone. But sometimes bacteria can cause a chronic infection with different organs and systems affected, and clinical presentations will be due to which organ or which system is affected. For treatment, again, antibiotics are essential in the case of moderate to severe cases. In mild illness, usually antibiotics are not required, but if you have some factors or some symptoms of chronic process, of course antibiotics are important. And for prevention, again, we have specific and non-specific measures. Of course, non-specific measures include proper handling of animals, proper cooking. If you need to remove some dead body or you're hunting on animals, it's better to use gloves and some mask. Proper sanitation of water in forest areas. If you went to some picnic or you have a trip, of course it's better to avoid drinking of water from natural sources. And protection against insects is a good measure for multiple disease, tularemia is included. So we will talk about clinical presentations in a separate manner, because actually it is a complex disease and have 6 major syndromes or 6 major clinical forms according to route of transmission. I didn't mention it before because for every type of transmission, for any way, we have a specific clinical syndrome or clinical form of the disease. So the most common type of disease is ulcer glandular, means that you have an ulcer in that subject, which is the gate of entry, and closest original lymph node is affected. Usually that type of disease is transmitted through some insects, means vector-borne way of transmission. It can be tick, it can be deer fly, it can be some type of mosquito, and that ulcer will be formed in the place of bite. Initially it is a simple vesicle after bite of the insect, but later it will be transformed to ulcer. It can be covered with some purulent film because of bacterial flora adjusted. And bacteria from that site of entry will migrate to regional lymph node and will cause regional lymphadenopathy. So please take a look on the left part of the slide. The picture is showing ulcer. Glandular form, next form is shown on the second slide. Usually in the ulcer glandular form, that lymphadenopathy is less severe. Inflamed, red, painful lymph node, but not such obvious as in the right part of the slide. Of course, general symptoms of infection can present, such as fever, malaise, headache, loss of powers, loss of appetite. So second form again is more severe and one of the most frequent types of the disease. It is a glandular form. When subject will have seriously injured lymph node, it can again, due to other bacterial flora or even caused by Francisella, it can separate, can form a lot of pus, and of course in that case surgical treatment is required and antibacterial treatment is required. Usually that form is transmitted through direct contact with animal or animal tissues. When someone touched that animal, or maybe some bacteria got into gloves or to the common wear, and when subject is scratching neck, some again mosquito or some fly near, you just mechanically or reflectively scratch. Bacteria is inoculated in the skin, and other parts of the body usually are closed, because in forests and especially hunters they wear camouflage and some other, and other parts of skin are closed, but face and neck are affected. So usually that form is located in axillary region because of direct contact with the fingers, or in the neck and face because of inoculation by hands of affected person itself. Third form is oculoglandular, means that eyes are affected, and again closest original lymph node is affected. Usually, again, that form is transmitted by direct inoculation with the hands when someone touched animal and scratched eye, or maybe by splashes. Diseases manifest as severe conjunctivitis. On the left part of the slide, you can see that eye with hyperinflamed blood vessels. They enlarged, they're very red with some hematomes. Of course, again, regional lymph node will be affected, and it will be obvious it will be severe injury because it is again head or neck lymph nodes. And common symptoms of infection will be present, such as high fever and malaise. Disease is dangerous because it can lead to loss of visual acuity and damage of some visual options, but usually it is transient, means after convalescence symptoms will disappear and vision will be back again. Fourth type is one of the most severe. It is oropharyngeal form. Usually it is observed after use of some water or food products contaminated with bacteria, and of course it is ingestion but not very deep when dose of bacteria is not high and main number of microbes will be absorbed in oral cavity. That form is manifest with severe headache. Both glands and both sides will be affected, so tonsillitis very serious will be observed, and it can lead to formation of exudative pharyngitis with difficult to breathe and some respiratory symptoms. So again, in the right part of the slide, you can see that inflamed affected mucous membranes of oral cavity and some films formed. Not very good, but it's difficult to make a good photo in oral cavity due to some obvious reasons. Two last forms are associated with most high mortality rate. They're systemic or generalized forms. Fifth one is typhoidal. Again, it is due to ingestion of water or food, but in large amounts, when microbe will be delivered to stomach and intestine and will be absorbed into systemic blood circulation. Disease will manifest with very acute onset of infectious symptoms such as fever, chills, severe headache, vomiting, tenderness and other. And diarrhea and some other GIT disorders usually a prominent manifestation of the disease. Stool is watery without blood. Some mucus can present, and symptoms will depend on dose of bacteria which was ingested. Sixth clinical form is pneumonic. It may occur as the last stage or most severe stage of or any of the previous forms, or usually is observed after inhalation of some dust from fur of dead animals, or inhalation of bacteria in the type of an aerosol. Again, different clinical forms are possible according to dose which was inhaled. It can be single lobular or multiple lobes can be involved. It can manifest with different X-ray findings. Usually it is formation of pneumonic infiltrates, or they're looking like an abscess. Lymph nodes, of course, will be affected, and you can see enlargement of hilar lymph nodes. Diseases, systemic disease will manifest with common symptoms such as fever, difficulty of breathing, pain in the chest, and of course loss of power, some headache and other symptoms. Of course, disease is extremely important to recognize as soon as possible. Disease, especially in some inhalation form, can be transmitted from human to human, or some excrements of the human can be infectious for subject, and of course it is important to isolate person in infectious department or infectious hospital or any other separate wire box. And of course it's essential to perform fast and rapid diagnosis. Again, we have the Tularensis biological preparation, which is actually an allergen of Francisella tularensis cells, and it is used for application on skin to identify disease, or if required to perform some vaccination in healthy person than to evaluate immunity. And the same tularemia diagnosticum, which is actually killed cells of Francisella tularensis, preserved in formalin. It is used as antigen to identify presence of antibodies in patient's sera. Of course, you should understand that in the first days of disease, antibodies cannot present in patient's sera, so that test is only for confirmation of immunity or for later diagnosis. So we have several types of vaccines against tularemia, which are still in research and development stage. We have killed vaccines, some subunit vaccines and recombinant vaccines, but no vaccine is available till the moment worldwide as some approved and confirmed safe vaccine. Historically, we had alive vaccines against tularemia, and the very first vaccine was designed and produced in Soviet Union and closer to the middle of the 19th century, and it was a very good vaccine with very good effect, and millions of people in USSR were vaccinated in endemic areas who worked with animals or who just lived in animals. The vaccine was pretty fine, safe, and showed very good results. So after World War II, some strains of Francisella tularensis were transferred to USA to the Army Medical Research Institute of Infectious Disease, which is located in Maryland and Fort Detrick, one of the most famous US Army's places. And actually that vaccine was transmitted from Gamaleya Institute, which is famous now because of that coronavirus vaccine. And again, it is some doubt. In many number of countries, that vaccine is not available. In Russia, we still have that vaccine. We're using it in endemic regions and in laboratory workers. Vaccine is still showing good results and showing good efficacy and safety. In USA and in many number of European countries, that vaccine was available, but recently it was removed from scheduled vaccination in groups of risk because of some safety doubts. It is still a live vaccine, and it is a doubt that that vaccine can be, strain of that bacteria can be used by some extreme organizations to induce a mutation and obtain again virulent vaccine. First of all. Second, some safety requirements are doubtful of that vaccine. It can cause some adverse events, and it's not available in USA and in many number of European countries, but again, we still have it in Russia. So, as a live vaccine of very dangerous and potentially infectious bacteria, vaccine is contraindicated or prohibited in children 7 years and younger. For adults and children above 7 years, we have only single dose, which is sufficient to induce good and strong immune response. And if it is required, if some subjects still have a risk factor to be infected by tularemia, booster dose is required every 5 years after initial first dose. So bacteria is dangerous and it is alive, so immunity is pretty strong.